This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Actually, hear me? Okay. So I wasn't really sure about what to share with you today, and I think I decided to offer you a sort of broad overview of my research topic with some glimpses of different studies rather than focusing too much on the details of the single experiment. And I hope it's fine and you will find it enjoyable somehow. So my research topic is clearly voluntary actions, and that's because voluntary actions are amazing features of human mind, but quite surprisingly, the experimental psychology is almost completely silent about voluntary action. So there is a sort of negative space in the literature all around voluntary actions. And the reason is that it's very hard to study voluntary actions in the lab setting. So it's a bit of a challenge, but it's an important one to me at least. And my research is trying to take some very modest steps in understanding the neurocognitive processes of voluntary actions. And I think that the first challenge that people studying voluntary action face when they start is that cognitive neuroscience doesn't really provide any satisfactory account of what a voluntary action actually is. And my approach to this question is to try and identify some features of voluntary actions, or better, some features of actions in general that can make some actions more voluntary than others. And probably neither of these features are sufficient on its own to make an action voluntary, but definitely some of them are necessary. So what I'm going to do today is to unpack those 4 different features and present some of the work that I've done in my journey of understanding voluntary actions. So the traditional approach to voluntary actions has typically defined voluntary actions by contrasting them with stimulus-driven actions. So imagine a continuum. At the end of one continuum, we have the stimulus-driven actions where the form of the action is directly determined by the form of the stimulation. At the opposite side of the continuum, we have these intention-driven actions where the form, the timing, and the occurrence of the action is not directly determined by any identifiable stimulation, at least. And this operationalization has turned out to be very useful in identifying what we call neural markers of voluntary actions. And these are typically brain regions or processes that have been constantly but not exclusively clearly associated with intention-driven behavior. And probably one of the most important one is the activity of the mesial wall of the frontal cortex. And this is evident in very early day studies from animal lesion experiments, for example, Vassingham, 1987, but also confirmed by more recent neuroimaging studies on humans. So for example, this is an experiment that I will talk more in detail later, but you can see here the results from a meta-analysis of the literature based on all the neuroimaging studies on intention-driven behavior. And you can see clearly the activity of this mesial wall of the frontal cortex and another cluster in the parietal cortex as well. However, this operationalization between stimulus-driven actions and intention-driven actions sounds a bit simplistic, and that's why in 2008, Brass and Haggard published this opinion paper that is what were the model of intentional action in the brain. And in this paper, they suggested that intention-driven behavior can be thought as a sort of special form of decision-making. And it's special because compared to perceptual decision-making, when the information is already there in the stimulus and the brain just needs to process the information, here the brain needs to internally generate some information. However, it's not that simple because they suggested that there are at least 3 different kinds of information generation for these intentional action decisions. And these are a decision about which action to make, a decision about when to make it, and a decision about whether to actually put the action in place or not. And they suggested that these 3 different components of voluntary actions can actually be represented in different regions of the cortex. And that was my first bachelor project. So I performed this meta-analytical review of the neuroimaging literature on intention-driven behavior. And the idea was to see whether it was actually possible to tease apart these three different components and look for different brain correlates for each of them. So I went through all the studies and I classified them depending on the adapted paradigm. And here you can see the results. So there's this very pretty, gradient in the mesial frontal cortex with the pre-supplementary motor area associated with the decision about when to act. The middle frontal gyrus and middle cingulate associated with the decision about which action to make. And this very rostral region here in the cingulate cortex associated with the decision about whether to actually put the action in place or not. However, at this point, I really wanted to run my own fMRI study because these three components had never been studied in a single experimental design with the same sample of participants. So I managed somehow to convince someone to let me use the fMRI for my master project, and I designed these experiments. So there are three different blocks, a what, one, and a whether block. So in the what block, participants freely decided whether they wanted to move their index or middle finger. In the one block, they decided whether they wanted to move the finger immediately after the cue or delay the answer a couple of seconds. And in the whether block, they either decided to plan and then put the movement in place, or plan the movement but then put a veto and abort the movement and not to act at all. Clearly, I also had a stimulus-driven condition in which participants simply follow the instructions. And here you can see the results from the interaction. So this is an interaction effect. So those are the brain areas that are most strongly activated for one component compared to the others. And again, this very pretty gradient. So you can see that the decision about when to act is located more posteriorly, the decision about when to act is halfway, and the decision about whether to act or not is located in the more anterior regions of the cortex, spanning also into the white matter. So just to recap the take-home messages from the analysis of this first component of voluntary actions, it looks like we can think about intention-driven actions as a form of decision-making, but it's special somehow because it's based on internally generated information. But the internally generated information is not really a unitary concept because actually there are different kinds of information, and we isolated at least three of them that are processed in different regions of the brain. But all of these components are somehow located in the mesial frontal cortex. So, second feature or approach to volition that I would like to talk about today is based on this comparison between planned, deliberate actions and random movements. And these latter are, they require any minimum cognitive involvement. And there is something on this comparison on the readiness potential literature. So the readiness potential is another very famous marker of voluntary action, and for the people who doesn't really know what the readiness potential is, the readiness potential is a gradual increase in negativity starting 1 second or 2 before the time of the action, and it continues to rise until the movement onset. At that time, there is a steep change into the polarity. And we know that the readiness potential is generated by the pre-supplementary motor area, so again, mesial frontal cortex, but we don't really know what the readiness potential represents. So according to a very influential theory at the moment, I'm not sure I agree with that, the readiness potential wouldn't be nothing more than just accumulation of noise. So this is a leaky stochastic accumulation of some unknown noise. But there are some early evidence from actually 1983 suggesting that the readiness potential is actually higher and began earlier when participants reported they have somehow pre-planned the movement compared to when they just acted randomly or capriciously, as Libet said. So I was looking for a way to capture this comparison in the lab setting, so with an experimental design that was not really based on self-report. And I figured there is one situation in our everyday life in which our action starts being completely random and then progressively they change and they are more deliberate and pre-planned. And this is when we are learning something new, I guess. So when you are learning a new skill, at the beginning you just try it out, so you try and you act by trial and error, but then progressively you adjust your motor plan until you reach a stable performance. So I wanted to capture this sort of progressive learning and transition from random actions to deliberate actions. And so I designed this temporal reinforcement learning task. So here participants needed to learn the correct time to act based on feedback. And the cover stories treated participants as farmers, so they had to plant the seed at the beginning of the trial, and the task was to wait until the fruit was ready and then press a button to harvest it from the ground. So they progressively changed their behavior. At the beginning of the block, you can see that they were acting completely randomly, but then, based on the feedback, they managed to reach a stable performance. So you can see here that the distance from the target time in the different blocks progressively decrease and converge towards an optimal waiting time. And you can also see that the trial-to-trial change, that is a measure of the update from one trial to the other, also progressively decrease. But probably the more interesting result here is that the readiness potential slope, in particular, increased with learning. So late in learning, when participants actually had an internal model of the correct policy, the readiness potential was steeper, growing faster. While at the beginning of the block, when participants were just randomly acting and they didn't really know what was the best policy to put in place, the readiness potential slope was very flat. And this somehow confirmed the very early evidence from 1983. But at this point, I wanted to play a little bit with the stochastic model from Aaron Sugar and try to see whether it was possible, and I'm not sure, so these are very, very preliminary results. But the idea is that learning could actually change the process of noise accumulation, and the way I did it was to change the leak parameter. So the idea is that learning may reduce the leak of the accumulation of noise, but we still need to fit the data and see what happens. But the messages here are that pre-planned action seems to be preceded by greater readiness potential than random movements. And if we accept somehow the readiness potential as a sort of marker and readout of voluntary processes, what we can say is that the neural processes of volition are actually stronger when you have an internal model of what to do or when to do in this particular case. So, third component. Literature on voluntary action has often been criticized because the actions that we use in our experimental paradigms are completely arbitrary and meaningless. For example, if you think about my traffic light experiments, people don't really have a reason to press a button with the right index finger or the left index finger. It's just completely random. While usually voluntary actions in our everyday life are meaningful and we act for a reason. So, is this what we really mean for voluntary action? Probably not. But how can we make our voluntary actions in our lab setting more meaningful? It's tricky because there are some very early, kind of 2 years ago, but attempts in which they played with the action value. But to be honest, this is not really the direction I want to take, because I feel like we are missing out the intention-driven component here. This task seems to me very stimulus-driven. Instead, my personal attempt of making voluntary actions somehow enriched and more meaningful is to adapt voluntary actions that are clearly goal-directed. And one possible way of doing this is to adopt a problem-solving approach. And why problem-solving approach? So I'm not sure if you're familiar with the Tower of London task, but basically participants need to rearrange these colorful balls between pegs to match a start configuration and the goal configuration within the minimum number of moves. So it's important that they pre-plan the sequence in advance. And actions in this Tower of London task, but it's probably true for other problem-solving paradigms, are clearly internally generated because, of course, you have the stimuli on the screen, but at the same time, the stimuli don't provide you with the solution, so you need to internally generate some information. But at the same time, these actions are clearly goal-directed. So I feel that this experimental pattern would allow me to respect some of the internal generation criterion, but adding the goal-directed element that was missing in the literature. And at the same time, I got excited about the idea of studying voluntary action in sequences, so moving a little bit away from the idea that we need to study voluntary action as artificial units. So the first study I conducted was an fMRI study. So I compared Tower of London problems where participants planned and solved the problem through internally generated actions with a series of instructed movements of the same sequence length. So here, participants didn't really solve the problem because the solution was guided and given to them by these red squares. So I compared these two different conditions, and surprise, you can see again the network for internally generated behavior. So you can see the pre-supplementary motor area, the superior and inferior parietal lobule, that is extremely similar to the meta-analysis that I conducted and showed you a few slides ago. But of course, this network is a broad frontoparietal network, so this is not the only region involved. And at this point, I kind of got interested in exploring the connectivity patterns between these different clusters. And I got particularly excited about the idea of exploring different connectivity patterns for the planning and for the execution phase. Because this is something that you can do very easily with the Tower of London task, because as I mentioned, most of the planning happens before the first move of the sequence. And this is true for short sequence at least, and this is very clearly reflected into the reaction times. So I adopted a functional connectivity approach, and I used the pre-supplementary motor area as a seed, and I looked for possible differences in the connectivity patterns between the pre-supplementary motor area and all the other regions in the brain, spoiler. And so I looked for differences in the connectivity between planning and execution. So these are the regions that were most strongly connected to the pre-supplementary motor area in the planning phase. So you can see again the anterior and middle cingulate cortex and the superior frontal gyrus and also the inferior frontal gyrus. While these are the brain regions, actually the only brain region that is significantly connected to the pre-supplementary motor area in the execution phase. So this is probably more related to the monitoring of the performance rather than planning. But to me, it was nice that the very same pre-supplementary motor area that is supposed to be the generator of voluntary behavior actually interacts with different regions depending on whether we are in the planning phase or execution phase. But of course, fMRI doesn't really have the temporal resolution for a fine-grained analysis of the temporal dynamics leading up to the time of the action execution. So I moved to an EEG experiment and I adopted a Vaupier approach that I'm not going to describe because I kind of know it already. But what I wanted to see was whether it was possible, based on the EEG activity preceding the action 1 second, to disentangle between intention-driven behavior and externally driven movements. And it looks possible indeed. So in pink you can see the areas in which the classification accuracy was significant. And to me, the nice thing is that actually this classification accuracy starts decreasing 250 milliseconds before the time of the action execution. And this is somehow compatible with the very, very old idea of the final common path. So the idea that at a certain point, the information needs to converge somewhere. And probably it's the primary motor cortex, and probably at this time, it doesn't really matter anymore whether the action was internally generated or externally triggered, because the action that has been executed is exactly the same. And it's also nice, probably, that so if you look at the topography here, you can see that the electrodes that contributed the most to the classification accuracy were actually the electrodes where usually we observe the readiness potential. That kind of makes sense. So take-home messages from this second component are that, first of all, goal-directed actions are associated with a very broad frontoparietal network, of course, but the connectivity patterns are very different depending on whether you are in the planning or execution phase. But also, we can actually decode the goal-directed action from simulative action up to a certain point, when you are too close to the action itself, it's no longer possible to say whether the action was internally generated or stimulus-driven. And at the moment, I'm using this experimental paradigm for other experiments, but I just wanted to offer you the very main result, because I kind of want to focus more on the last component. And I thought that was super late, but actually no, I'm going to be too fast, sorry. But maybe I can drink some water. And last component of voluntary action is about consciousness. So voluntary actions are accompanied by some subjective, distinctive conscious experiences. And there are at least two different conscious experiences that we can isolate. One is the conscious experience of intending, so planning or being about to do something. And this experience is usually studied using the Libet clock, and that's why I'm not going to do it. But there is another subjective experience of a voluntary action that is the later sense of agency, that is this feeling of controlling your own actions and through them the events in the external world. So more simply, sense of agency is the feeling of making something happen. And this is the main focus of my PhD, actually. And I've never presented this stuff because COVID happened, so this is kind of the first time. But we don't really have a good model of action control, and Carl was very, very happy with this model. But it's not my fault, it's not my model, so it's Blakemore and Fried 2002. But there are two important features of the sense of agency according to the comparator model. The first one, that is more a process rather than a feature, is the idea that you need to make some predictions. So when you're about to move, you make predictions about the possible sensory consequences of the movement. The second component is the comparison. So you need to compare your predictions with what actually happened. And according to this model, so not my fault, when there is a match between the predictions from the forward model and what actually happened, you have a strong sense of agency. When there is a mismatch between predictions and consequences, it's justified to experience a reduced or absent sense of agency. However, the neural basis of the sense of agency are very much unclear, and in particular, it's unclear whether we can find different neural correlates for the predictive process and the comparative process. And that was my obsession at the time. But of course, measuring the sense of agency is difficult because we're still playing with voluntary actions. And usually people ask directly participants, what's your sense of agency? Did you do that? But the question is a little bit odd because usually in our everyday life we don't really have a strong, explicit experience of agency. So our sense of agency is actually running in the backwards of our mind, and we are only somehow implicitly aware of it. So asking directly is a bit odd. And that's why we try to use implicit paradigms instead that are based on indirect measures of the sense of agency. And probably the most famous one is the intentional binding effect that has been introduced by Haggard in 2002. And what's the intentional binding effect? It's a bias in the perception whereby every time you make a voluntary action that is followed after a short delay by the consequence, you tend to perceive this action outcome delay as shorter than its real lasting. So there is a time compression here, and it's a sort of binding of the action and the consequence. And this has been suggested to be a good measure of the sense of agency because it happens only when you make voluntary actions, or even better, when you make action in an active mode. Because when someone presses your finger to produce the very same action, or as in the original paradigm, you use a TMS in the primary motor cortex to produce a passive movement, the effect is completely absent or even reverted. I never found the reverted effect, but it's actually not present. So what I wanted to do for my PhD was a complete mapping of the neural mechanism of the sense of agency using the intentional binding effect. So I designed this temporal judgment task with an active and passive condition. In the active condition, participants saw this green light bulb, and they were instructed to press a button to turn the light bulb on. And the light bulb went on after a variable delay that was 200, 400, or 600 milliseconds. And then I asked participants to judge the action outcome delay, so the time passed between their button press and the lighting on of the light bulb. The passive condition was exactly the same, apart that the light bulb was colored in red and participants were instructed to remain still because the experimenter, that was me at the time, pressed a button to turn the light bulb on. So same thing, then they were asked to judge the time delay between the passive action and the effect. And I computed this time compression, that is the difference between the estimated and the actual delay. So you want negative numbers here, because negative numbers represent underestimations of the delay, so the compression. An intentional binding effect is seen when this underestimation, so negative time compression, is greater in the active condition rather than in the passive condition. Otherwise, it's just a bias in time perception and is not really related to the agency. And with this experimental paradigm, I managed to see an intentional binding effect, so you can see that participants underestimated the action outcome delay more in the active condition compared to the passive condition. But quite surprisingly, the effect was significant only when the time delay between the action and the outcome was very short, so when there was some sort of temporal contingency between the action and the outcome. Because at the later delays, longer delays, the intentional binding effect was actually not significant. But so what I did was to focus on the intentional binding effect at 200 milliseconds of delay. And I performed a linear regression between the fitted board activity recording during the fMRI task and the time compression at 200 milliseconds of delay by subtracting the active and passive conditions. And you can see here that there was an association between frontoparietal network again that is extremely similar to the one that I showed you before, and the magnitude of the intentional binding effect. So the greater the intentional binding, the stronger the board activity in the parietal cortex and presupplementary motor area. But still, I wasn't very happy because I wanted to see whether it was possible to somehow associate different roles to these different nodes of the sense of agency network. So I performed two different transcranial magnetic stimulation experiments. In the first experiment, I applied online repetitive TMS before the action execution, where supposedly we are making some predictions about the consequences. So this is supposed to be the predictive phase of the sense of agency. In the second study, instead, I applied online simulation after the action execution when the comparison is supposed to be happen. And I put my coil on the presupplementary motor area and the parietal cortex. I also had an occipital control site using the single subject MRI where I mapped the MNI coordinate from the fMRI experiment. And this is the main result from the first study. So you can see that when I interfered with the activity of the presupplementary motor area at the time of, so before the action execution, at the predictive process time, the time window for the intentional binding effect expanded, because you can see in the baseline condition that I had the intentional binding effect only at 200 milliseconds of delay. That is exactly as in the fMRI experiment, but this is a new sample size. But after the simulation of the presupplementary motor area, the intentional binding effect was seen as 400 milliseconds delay as well. And we somehow interpret this result as a sort of an effect of a less precise predictive process. So I'm still discussing the result with Patrick after 3 years, and the idea is that if it's true that this is our predictive phase, interfering with the presupplementary motor area at this time should somehow interfere with the predictive process. So the prediction may be less precise, and that's why the time window for the intentional binding effect may be somehow more blurry. While stimulating at the time of the action consequence didn't interfere with intentional binding at all. So I will recap later, but what you need to know about this experiment, very briefly, probably not my best experiment, is that the coupling between intentional binding and the pre-supplementary motor area is still valid when you switch from a visual modality to an auditory modality. But the interesting thing is that it looks like so the intentional binding is somehow tuned on different time windows depending on the feature of the task itself. So in this task, it was an auditory task. I didn't have intentional binding at 200 milliseconds, but I had it at 400 milliseconds without TMS at all. And probably this is still due to the fact that we have different expectations that are somehow tuned to the specific features of the task. And that's why I do think that the delay between the action and the effect is a crucial component of our sense of agency experience, even though Patrick is not particularly happy about it. But my last PhD study, and it's actually the last thing that I'm going to present today, is this experiment in which I was interested in the sense of agency in Gilles de la Tourette patients. And Gilles de la Tourette patients, I'm not sure if you're familiar with that, but it's a childhood onset movement disorder that is characterized by unwanted hyperkinetic production, so tics, but also abnormal vocalizations. And the tics in Gilles de la Tourette patients are particularly interesting because the nature of the tics is not really clear. So we don't really know if they are voluntarily or involuntary. Clearly they are wanted, but at the same time, environmental factors like being engaged with other things or internal factors like anxiety can modulate the frequency of the tic and even suppress them at all. And the other interesting thing is that participants report a sort of urge to move, a sort of premonitory urge. So before the tic, they feel like they are about to do something. And that's the very same experience that Libet described in 1983. So that's why I was particularly fascinated by these patients before I discovered that they swear a lot to you. But I repeated the very same experiment in fMRI using the light bulb paradigm. And you can see that Tourette patients don't have an intentional binding effect. So the intentional binding is severely reduced and is actually not even significant. So the comparison between the active and the passive condition, there is no significant difference in their underestimation. And the nice thing, of course, I didn't have so many patients, so that's what I had. The greater the severity of the motor tics measured by the Yale Global Tic Severity Scale, the more reduced intentional binding effect. So it looks like there is a direct association between the impairment of the motor system and the sense of agency as measured by the intentional binding. This was associated with the hypoactivation of the pre-supplementary motor area, but also a complete absent correlation between the sense of agency network and the intentional binding effect. So to recap about this last component of voluntary actions, I think the sense of agency strongly depends on the coupling between action and effect, but with the crucial role of the time delay between these two events. And different expectations may actually play an important role in setting the prediction or expectation for the sense of agency to happen. And it looks like from my fMRI and TMS experiments, the pre-supplementary motor area again seems to be a likely area to take care of the predictive process. So this is kind of the end. So there are the 4 features of voluntary action that we described today. And I have 3 slides of acknowledgements now. University of Milano-Bicocca people, in particular my PhD supervisor Laura Zapparoli. Patrick, so there are no words to say thank you to Patrick, but Steve and Carl and the Action and Body Lab people as well. My friend Stefan, who invited me here, Action and Body Lab people, who showed me how not to do camping in my life ever again. And as Stefan briefly mentioned, I'm leaving ICN, sadly, not going too far away, just 10 minutes, and I'm starting my own lab in January 2024 at Birkbeck University. That is